Okay, we look pretty stable here with numbers, so we're going to get going. Hi, I'm Sarah Jandrisic. I'm the Greenhouse Floriculture IPM Specialist for OMAFA, and I'm here with Cassie Russell, our Nursery and Landscape Specialist as well. Cassie, wave to the nice people and say hi. And you're watching the Grow On webinar series, which aims to bring timely production and IPM information to greenhouse operators and also nursery crop operators sometimes. So today we're going to talk about nematodes. I'm going to introduce Greg Bryant now. He originally studied entomology at Auburn University and then was a research technician at North Carolina State and a pesticide company. I met him at NC State when I was doing my postdoc there. And then Greg started working in the biocontrol industry in around 2017. So we're glad to have him with us, and he's now the technical lead at Beneficial Insectary. So, Greg, I will let you take it away. Oh, one more housekeeping thing. I just wanted to mention if you're having any issues, put them in the chat and we can see if we can address those. And Cassie will be monitoring the chat. If you've got questions for the end of the webinar, we'd ask that you put those in the Q&A function. It's a little bit easier for us to keep track of it that way. Okay, so without further ado, I'm going to mute myself and hide my video. And Greg, you take it away.
All right, thanks, Sarah. So, all right, let's just jump right in. Let's see. All right, so we're just talking about entomopathogenic nematodes. So let's kind of go over just what they are to start with. So to break down the word entomo means relating to insects, and then pathogenic is disease causing. Might sound kind of odd that something would be pathogenic like this, but that is due to the symbiotic bacteria that they have that actually kills the pests. Overall, they're useful for a few hundred pest species, over 250. There's lots of different species of entomopathogenic nematodes, but the two most utilized genera are Steinernema and Heterohabditis. And then each genus of nematode has a specific genus of bacteria that is associated with them. So, just a bit about their life cycle and how they actually work. So the infective juvenile stage, they will actually wiggle their way into the pest, their host insect that they are trying to infect. And they can go in through the spiracles down the side. They can go through the front end, through the mouth, or through the back end. Just whichever opening they come to first is the one that they'll use. Once they're inside, they release that symbiotic bacteria that's been living in their digestive tract. And that bacteria is toxic to the host, and it actually kills them. It basically poisons the blood of the host insect. From there, the insect is going to take anywhere from about 24 to up to about 48 hours to actually die. Then the bacteria that was released proliferates. The nematodes start feeding on the bacteria that's breeding in there. The nematodes themselves breed, they lay eggs, they go through their whole life cycle. Then once that cadaver is kind of used up and no more bacteria can grow in it, more effective juveniles will kind of bust out and go in search of another host and do the whole thing over again. So that's kind of a cool life cycle and kind of interesting, but why actually use these nematodes? So they're really easy to incorporate into whatever kind of pest management program you're currently using. They're compatible with a lot of different pesticides that you may be using right now. So kind of a way to be proactive and not so reactive. So nematodes work best when you've got low levels of pests. You're just trying to not let them get um, get more out of hand. Uh, no pesticide resistance issues. So these insects cannot build resistance to being infected by these nematodes. And it's a way to extend the life of the chemistries that you do have because you're not having to use as many. I can save labor from the standpoint of not having any worries about REIs, no harvest intervals to worry about, and no risk to plant health. Very, very safe for plants. And they pretty much only go after the pests that you are using them for. There's not really any negative impacts on pollinators or other beneficials. And so when it comes to these entomopathogenic nematodes, there's two ways to produce them. You can produce them in vitro in these vats of this artificial diet, or you can produce them in vivo. And the most common way to produce them in vivo is in waxworm larvae. So with the in vitro method, you've got nematodes that end up with varying amounts of that bacteria per nematode. With the in vivo style of producing nematodes, pretty much every single nematode is going to be full of that symbiotic bacteria, very efficacious and just good at its job of eliminating pests. And so, what this means for growers is the rates that you use are very different. So, with the in vitro style production, since the nematodes have often varying amounts of that bacteria, you want to use 50 million per 1,100 square feet every 2 to 3 weeks, or you can use a higher rate, 50 million per 550 square feet or 50 square meters every 4 to 6 weeks. With the in vivo style production, you want to use 24 million per acre, or about 30 million per hectare as a prevention. And then as a curative application, 48 million per acre or about 60 million per hectare. Uh, the the cost per square foot ends up being about the same. So it's really just kind of user preference. And just to note, the in vitro produced nematodes, they tend to come in either a powder pack or like a gel pack, whereas the in vivo produced nematodes are sent on sponges. And there's no other carrier besides the sponge. When it comes to actually being a nematode, you've got two different styles. You can be a cruiser type of nematode that actively searches through the soil profile looking for pests. And they tend to live deeper down in the soil profile. You can also be an ambusher style nematode. They just sort of sit and wait for pests to pass by them. But anyway, getting back to HB nematodes. Like I was saying, they're a pure cruiser type of nematode. They like to live deep within the soil profile. So when you water them in, you want to water them in pretty heavily, but not to run off because that means that you're pushing them all the way through. Useful for sedentary pests because they actively search them out and go find, and they go to the pest themselves. Useful for a lot of types of beetle larvae, including black vine weevils. They are a warmer weather species of nematode, so their optimal soil temps are anywhere from about 70 to 85 Fahrenheit or 21 to 29 degrees Celsius. Now we got Styrenium acrosii. They're also cruisers that live deep within the soil profile. Again, you want to also water them in heavily, but not to the point that you've got runoff. The advantage of Crassi is they're uh, have a wide range of temperatures, and they're very cold tolerant compared to most other nematodes. So they're useful anywhere from 41 to about 70 degrees Fahrenheit or 5 to 21 degrees Celsius. The most useful application that they have and what the most commonly used pests for them are black vine weevils in cooler weather, so like very early in the spring or later in the fall. We got Styrium carpocapsae. They are pure ambusher, so they don't actively search for pests. They sit and wait for uh, whatever larvae they're wanting to infect to actually come to them. They like to live on the surface of the potting media, or just below it. So when you water them in, you want to use as little water volume as possible. Make sure that your plants are pretty well irrigated already, and then just very lightly mist or just lightly water them on to get them right there on that soil surface. And oftentimes you can see it's called nictating. It's a little behavior that they have where they wiggle around. They kind of attach themselves to a piece of soil on their back end and then wave their face around, just waiting for a pest to come by them. And once they detect something that they might want to infect, a lot of times they'll kind of put their face towards their back end and fling themselves at it. It's kind of fun to watch. So they are mostly good for mobile pests because, like I said, they are just a pure ambusher, just sit and wait. In the greenhouse, most of the time they're used for shore flies. Also useful for certain beetle larvae, including black vine weevils and red-headed flea beetles. Certain caterpillars can be hosts for them, leaf miners, and then in yards and turf, chinch bugs and fleas. They're also useful for. Now, their optimal temperature range is anywhere from 60 to about 85 or 15 to 29 degrees Celsius. But with carpet capsa, you've really also got to factor in air temperature because they are really living on the surface of the potting media. We got starting name of Feltier. They can do a bit of both strategies. They can cruise a little bit. They can also do some ambushing, do some of both behavior. They're an intermediate forager, so that means that they don't live quite on the surface, but they don't like to be very much below the surface either. So about an inch or two or 3 to 5 centimeters below the surface is where you'll typically find starting name of Feltier and where you want to get them when you're watering them in. So what I like to have people do with Feltier is water their plants to maybe about 75% of their usual capacity with no nematodes. And then with that last 25%, put the nematodes on. And that tends to get them right there in that top level, like right where they want to be. Most of the time in greenhouses, they're used for fungus gnats and pupating thrips. They're cooler to moderately temperatured nematodes, so anywhere from 55 to 80 degrees Fahrenheit or 13 to 27 degrees Celsius is just right for them. So just to go over a bit of a visual representation of these nematodes and their foraging strategies and where they can be found. So again, we got starting Carpet capsae. They like to be right there on that soil surface. Going to infect mostly mobile pests that are going to come to them. We got Feltier that's going to live, do that intermediate strategy where they're kind of near the soil surface, but not quite on top of it. Then we've got the two pure cruisers, Starnum acrosii and HB, live very deep down in the soil. They're going after the more sedentary pests. So let's talk about some of the specific pests that we're going after. So fungus gnats is a big one. The adults kind of look like mosquitoes a little bit, not very strong flyers. You want to look for long antennae and long legs. But the most useful characteristic, if they're attached to a sticky card in a certain orientation where you can see that wing venation, you can see that very characteristic forked or Y-shaped wing vein right there in the middle. If you see that, that's going to be a fungus gnat pretty much every time. Their larvae are white and they got this shiny black head. And their larvae thrive with high moisture levels in the potting media. So one of the best things you can do for fungus gnats is really just back off on watering it, if possible, and dry down that potting media. So the life cycle, the biggest point to pay attention to with the life cycle is the larvae and how long the larvae live. So the larvae are going to live for anywhere between 5 and 14 days. I know it's a pretty big window, but it's so temperature dependent. Cooler weather, it's going to be closer to 5 days, but if it's pretty warm, or I'm sorry, backwards. If it's pretty warm, they can complete that larval cycle in about 5 days. But in cooler weather, it's going to take them up to a couple of weeks. But that is the only window that you've got where Styrene FLTA or HB nematodes are going to work because they're only going after that larval stage. So for preventive applications, you want to treat young plants that you just stuck or seeded within about 2 to 3 days, and then really just depends on the pest pressure. You can repeat for prevention every 7 to 14 days, or really just as needed based on pest pressure. In situations like propagation, where the potting media is just constantly being misted and everything's very wet, obviously you're going to want to go on the shorter end of that range. For curative applications, you really want to try to break that life cycle. So make at least 2 to 3 applications about 5 to 7 days apart, and hitting them with that overlapping generation with the nematodes a couple of times really tends to break that life cycle. Stein and Mepheltiae are usually the go-to for fungus gnats, but if you're growing in deep containers, and especially if they've got drainage holes at the bottom where the fungus gnats can lay their eggs, And if you've got fungus gnat larvae kind of all through that soil profile, the HB nematodes can be very useful in those situations because obviously they're going to live much deeper down than the Feltier will. And so for short flies, they look more like small house flies, not very mosquito-like, like fungus gnats are. Um, very strong flyers, especially when you compare them to fungus gnats. They've got short antennae just like houseflies do, but the main thing to look out for with short flies is those five white spots on the wings. If you see that, you got a short fly every time. Their larvae look similar to fungus gnat larvae, but a little darker, a little more cream colored or brown, but they don't have a visible head like fungus gnat larvae do. And since they actually require algae, they need a lot higher moisture content than fungus gnats. So I normally see short flies in more like hydroponic growing situations. It's where they tend to be the biggest problem from the growers that I work with anyway. Their life cycle, the adults can live a lot longer than fungus gnats, and their entire juvenile cycle is much faster. So the larvae only feed for 3 to 6 days before pupating. Um, and so you've got a much shorter window of where nematodes are effective for them. So they can be a little bit harder to control for that reason. The larvae are typically found closer to the surface where algae is growing, or in puddles or just anywhere else that you've got algae build up. Um, So for nematode application, again, just kind of as needed, but you do want to bump that window up if you've got high pest pressure. So instead of every week or two, make that every week to about 10 days because of that shorter life cycle for the juveniles. Curative applications can be about the same, so 2 to 3 applications, 5 to 7 days apart. But the biggest thing for short flies is you've got to control that algae. If you've got puddles, if you've got standing water, if there's algae growing on your pots, you've got to control that. If you don't control the algae, even with applying nematodes, you've got almost no shot at controlling them. And Cyanoderma carpocapsae are by far the most effective for short fly control because they tend to be found in the same areas. So those short flies are going to be living, like I said, mainly on the surface of the pots where that algae is growing, and carpocapsae also likes to live there. So I know we have a few nursery growers here, so I wanted to talk a bit about black vine weevils. The adults, kind of grayish black. They've got a bunch of ridges down their abdomen, and then they've got these tufts of hairs that give them looks like little white or yellow marks on them as well. They're weevils, so they got a little short snout on them. And their wing covers are actually fused, so they can't fly like most of the other weevils can. But they are still highly mobile. They're very good at crawling around and climbing things, so they can definitely get around even though they can't fly. Every single black vine weevil that exists is a female. Just males are not known to occur, so they reproduce via parthenogenesis. So females basically just clone themselves and lay eggs. The larvae, they're kind of cream colored. They have no legs, and when they're at rest, they make a little C shape. So they're actually going to overwinter in nursery stock as older larvae or pre-pupae in the soil. Um, they can overwinter as adults occasionally if they've got shelter. So either in leaf litter, sometimes in structures. But the vast majority of the adults are actually going to emerge in the spring, and they've got to feed for a few weeks before they can start to lay eggs. And then each adult lays anywhere from 200 up to about 500 eggs. 200 to 300 is most common, though. The eggs are laid kind of close to their host plant, take a few weeks to hatch, 2 to 3 weeks. And normally they only have one generation per year, but in protected environments, they can have multiple. So when to apply nematodes? You've basically got two windows for black vine weevils. March through May, when those overwintering larvae are waking up and starting to feed. So if you apply nematodes during that time, before they actually turn into adults, you can reduce the populations there. And then also, once all of those eggs have been laid throughout the summer, you can apply nematodes again, anywhere from typically August through November. And Um, in the spring, you actually might need repeat applications just depending on pest pressure. And then, of course, if you get any new plant material in that you suspect may have black vine weevil larvae, go ahead and treat those new plants as well. And the good thing about black vine weevils is nematodes can infect both the larval and pupal stages. They are susceptible to multiple different kinds of nematodes. So HB nematodes, Kraussii, or Carpocapsae are all effective. The one that you pick, base it on temperature. So if it is in cooler weather, definitely go for Kraussii. If it's kind of moderate, cool to warm, the HB nematodes are good. If it's on the warmer side, then go for Carpocapsae. So, also want to talk a bit about red-headed flea beetles for the nursery folks out there, and some of the ornamental folks see them occasionally. So the adults, they are very shiny with a red to kind of brownish head. Their hind femurs are enlarged, and they definitely do not skip leg day. They can jump pretty far with those enlarged hind femurs. Uh, the damage that they cause is called shotgun hole damage. So looks just tiny holes all throughout the foliage. So definitely makes things ornamental plants ugly and not easily sold in that condition. The larvae are kind of thin. They're elongated, kind of a creamy white color. And they've got three pairs of legs, kind of close to their head. They do feed on roots, but it normally doesn't really damage ornamentals. Um, So but obviously, killing the larvae can reduce that adult population that's going to be damaging the foliage. And so, for their life cycle, they tend to overwinter as eggs, and growing degree days is a big point of emphasis for red-headed flea beetles. So, 250 growing degree days with the base 50 is when they can first be found. The first generation larvae, they're going to be emerging in the spring, and you can find them up to 800 degree days with the base 50. The larvae are going to feed for quite a few weeks before they pupate, so pretty long window to get them with nematodes. So if you've got plants that were infested the previous season, it's definitely a good idea to start scouting them early in the spring when you hit that degree day threshold. And then apply nematodes once those larvae have been found. And Carpocapsae have been shown to be by far the most effective. And just for some scouting tips, you want to scout on warm days because if it's too cold, they'll be the most of the larvae are going to be pretty much deep inside that root ball, and you'll never find them on the outside. And you want to do that about half an hour to 1 hour after irrigating, because if the outside of the root ball is too dry, they're going to be more towards the middle. And the larvae also don't like light, so scout either on cloudy days or you can actually shield that root ball just with your body from the sun so that they're not getting light shine on them, and that helps you find them better. So just kind of a quick recap of the pests and the nematodes that go after them. So fungus gnats, you can use Styrianema feltiae or Heterorhabditis. With the shore flies, pretty much just Carpocapsae. Thrips, assuming it's a species that pupates from the soil, such as western flower thrips or onion thrips. Both Feltiae and Carpocapsae can help. Black vine weevils, you got a few options, HB, Kraussii, or Carpocapsae. Feltiae, they can affect black vine weevils, but from the research, it's just they're not the most effective at all. So definitely stick with one of those other three species. For the red-headed flea beetles, you pretty much want to only be using Carpocapsae. All right, so you've got these nematodes in, you're all ready to use them, but we got to make sure that they're alive. So to do some QC on them, put a small amount just in a clear dish, like a petri dish, or just any other kind of shallow container that's clear. Put a dark background behind it, shine a light on top, and just look for movement. If the nematodes are dead, they're straight like little sticks. If they're wiggly and curly, that means that they're happy little nematodes and going to be looking for pests. Best to do this before and after spraying, just so that you know that, hey, they're alive before I sprayed them, just in case anything weird happens in the spraying process. You want to make sure that they're alive after that spray as well. So these are all effective juveniles. So if they have kind of a dense appearance, they tend to have better infectivity. If they're kind of clear or transparent, they can still be alive, but they may not be quite as infective. Very important to note if you are QCing in vivo produced nematodes, use a super, super thin layer of water. And the reason to use a thin layer of water is when the nematodes are harvested uh, with the in vivo style, a bunch of other stages of nematodes besides the infected juvenile stage are also going to fall out and be collected. No other nematode stage can be free living except the infected juvenile stage. So all of those other stages are going to die. Obviously, the companies that produce in vivo nematodes know this, so they take that into account. And whatever amount of nematodes that they say that they're sending you, those are just the effective juveniles. They don't count the dead ones, obviously. But the reason that this is important for QC is dead nematodes float, but living nematodes sink. So if you've got too thick of a layer of water, all you'll see are the floating dead ones on top, and you'll think, hey, I would just send a bunch of dead nematodes, but all the live ones are at the bottom. So use a super thin layer, and you'll definitely see those living and wiggly nematodes. With the in vitro style, so the ones in a powder or a gel pack, they only send the infected juveniles, so you don't have to worry about that thin layer of water. So for your application equipment, just make sure that your nozzles are large enough. Take out any filters that are 50 mesh or smaller. Make sure that the PSI stays under 300. Under 200 is best, but they can go up to about 300. Um, they can go through boom systems, but again, remove every single one of those little filters. You can use backpack sprayers, but it's best to just use like a hose-in sprayer and just drench them in that way. So you also want to make sure that these nematodes are agitated in the tank. If your outtake valve is at the bottom and the nematodes aren't agitated, they'll all sink, and then you'll be putting out a bunch of nematodes right at the beginning, and then kind of hardly any towards the end. So you can do that with air stones or air discs with an external air pump with the line hooked to it. You can also use internal pumps. Research has shown that internal pumps can actually kind of mechanically beat up the nematodes a bit and maybe heat up the water a little bit too much. So you can have some loss of, you can lose some of the nematodes that way. So using an air stone is better, but it's not going to be too big of a deal to use an internal pump as well. Lots of people do that with no issues, but if you're just choosing one or the other, I would go for the air stone idea. Dram actually makes buckets that have aeration and agitation kind of built in. If you want to go that route, they also, if you're doing bigger applications, they've got verticarts with 50 gallon. There are 50 gallons, and they also have aeration and agitation built in. So you can use nematodes through irrigation lines, but it can be a little bit tricky. So just make sure that the nematodes are okay. So QC them after you put them through those lines. So the reason that it can be detrimental to them is if there's dips in the lines, or if the water just doesn't move quickly enough through those lines, the nematodes can bunch up, and you get uneven application that way. Sometimes there's just not enough oxygen in those lines, and they'll die due to lack of oxygen. Sometimes the pressure just doesn't push them all the way to the ends of the lines, so you get a bunch of nematodes starting out, and then they taper off the further from the origin point you get. And also right where those those lines are put into the pots, that's pretty much only where you're going to have the nematodes. And ideally, you would have them covering the entire surface and evenly dispersed that way. So it can be done. There's just quite a few things to look out for. And like I said, definitely capture some nematodes that you're putting through those lines. Put them um, capture them at the ends, and just make sure that they're alive and well, and they're actually making it all the way there. So for the application itself, if the nematodes are stored in the refrigerator, let them warm up to our room temperature for about half an hour. Because if they're going from a really cold fridge into warmer water, that can give them heat shock. You want the water temperature to be fairly warm, 60 to about 75 or 16 to 26 C. Fill up the tank about halfway, start the agitation, then mix the nematodes in, put the rest of the water in there. And then again, just make sure that that mixture is continuously circulating, because if not, nematodes can drown. Takes a while, but they are not aquatic animals, so they're not meant to be in water like that. So you do want to use them up within about 2 hours. To store them, just keep them in the fridge. And keeping them in the butter compartment of a fridge actually tends to keep them at the perfect temperature. So ideally, you would have them around 38 to 41 degrees Celsius or Fahrenheit, or about 3 to 5 degrees Celsius. The in vitro style nematodes have preservatives in the carrier, so there's going to be an actual expiration date on those packages. The in vivo style nematodes, they don't have any preservatives, so there's no actual expiration date. They should last at least a couple few weeks in the fridge, but definitely make sure that they pass the smell test before you use them. Even living nematodes don't smell like roses, but they should not make you want to revisit your breakfast. Always make sure to keep EPNs out of sunlight because UV is very harmful to them. So best to apply them either very early in the morning or after dusk or on a really overcast day. They can go into diluted fertilizer solution, but do not put them in your stock solution because that is too salty for them. You can tank mix them with compatible pesticides, and I like to have a lot of my ornamental growers dip their young plants in a mixture of Bavaria bassiana, so something like Principal from Bioworks, Trichoderma, something like Root Shield, and Nematodes. So just kind of sum everything up. Very complex life cycles, but I find it really interesting. Definitely follow the recommended rates for either in vitro or in vivo style produced nematodes. Just keep in mind how different those rates are. QC them to make sure they're alive, and do that before and after you apply them. Definitely use the right application strategies and the right equipment. And then keep in mind the foraging strategies for the different nematodes. Use the right species for the pests you're going after. And one of the biggest things is just get each species just where they'll be most effective. And that's about all I got.
Amazing. Thanks so much, Greg. Maybe stop sharing your screen and we'll just go to the question and answer period. But first of all, I just wanted to thank you for surviving your first webinar. Even with technical difficulties, I feel like that's part of just like the trial by fire that is doing webinars nowadays.
Yep.
So with that, Cassie's got some questions that were appeared in the chat.
Yeah, so just to encourage, if you do have questions, please put them in the Q&A box. We do have a few there, so we'll go through those. So first, there's a question here. How deep is deep when it comes to the nematodes living deep within the soil?
So they'll go pretty much wherever the pests are. The HB nematodes and the Kraussii. Um, I don't know what their limit is, to be honest, but for the typical pest that we're talking about, that's where they want to be as well. So that's really the main point of that was to just get them where those pests are.
We have another question. How effective are EPN nematodes when applied as a preventative control on newly planted stock? So how long will they live in soilless mix before having food?
So it all depends on their lipid reserves. So the in vivo style nematodes tend to have larger lipid reserves than the in vitro style. So they can persist longer in the soil, but environmental conditions play such a huge role in it. The warmer it is, the faster they're going to use those lipid supplies up. You've also got to take into account moisture levels. So if it dries out, nematodes are extremely sensitive to desiccation. So in the correct environmental conditions, if it's cool enough that they're not going to use up those liquid reserves, and if it is if the moisture levels are adequate, nematodes can survive for weeks, if not months, in the soil. How effective they're going to be, though, that really depends on those environmental factors.
Awesome. How many days can say steer oh gosh, I'm so bad at pronouncing these names. SF nematodes in the soil. How many days can they live in the soil waiting for fungus gnats? How many days or weeks can they live? Can live SF complete the cycle? Sorry, a little bit of a repetitive question there.
Yeah, basically same answer. So
Yeah.
it just depends on those environmental factors mostly. And then several generations of nematodes can be completed within that insect cadaver before it's used up.
All right, a little bit of a different question. What about what new nematode? There's something about a new nematode for the larva of Thrips parvis finus. Do you know anything about that?
Not that I know of, that might be more of a fair
Maybe
question.
there.
Yeah, I don't know if there's a new nematode, but I know that Alexandra Ravinthi in Florida did do some screening of the commercial nematodes for Parvis finus, and basically Feltii didn't do well even in the lab trial, so they didn't move on to the um, full plant trials in the greenhouse. So they found more it was like the Carpocapsae and the Heterorhabditis species that did better. She just published a paper on that. Maybe I'll post that on the blog. I'm not sure if it's open access or not, so if I'll link it on the blog so you guys can read it.
It's interesting to me that the surface species and the deep species were good, but the intermediate species not so much.
Yeah, I have no idea. So yeah, I need to read more of that paper to find out. But it kind of does make sense that different thrips species maybe are susceptible to different nematodes. Who knows?
Sure.
All right. Another question here. So you were writing that SC work against thrips, but only the soil living stages. What do you think about Copert's claims they work well against all kinds of stages on the plant?
So yeah, so nematodes can definitely infect the larval stage or the adult stage. SF and SC can. Um, Personally, I don't love using nematodes on the foliage. It can cause other problems because you've got to keep that foliage wet for a while, um, and nobody wants wet foliage. Just for disease issues. So unless the nematodes have a water pathway to swim through to get to the pests, they're not going to find them, or they're not going to be able to reach them. So yeah, like you can on a very cloudy day or where you're not going to have a lot of UV, you can use nematodes on the foliage and then in about half an hour kind of mist the plants to keep those water levels up. But um, most of the time, I personally don't think that it's worth it.
Makes sense. Have you trialed SF nematodes for shorefly larvae?
So they will infect shorefly larvae. They're just not typically found in the same strata in the soil. So if an SF nematode comes in contact with the shorefly larva, sure, it's going to infect it. But SF like to live kind of below the surface. But like I was saying, the shorefly larvae they're so reliant on algae, and algae is pretty much only going to grow on the surface. So that's where the shorefly larvae are going to be.
Okay, and then we have a couple questions here about kind of application. So we'll jump to those. What's the most effective application method for both the cruiser and ambushers?
Um, well, so again, just water the cruisers in kind of hard and heavy because you want to get them deep down in the soil profile. And then even about 20 minutes after you water them in, about half an hour or so, irrigate again just to get them even deeper down and just make sure those moisture levels are still really high. And then the other question was about the ambushers. So I like to have the potting media irrigated pretty well, almost to full capacity, and then super lightly water them on because you really just want them to be sitting there on that surface.
All right. And then again, on the equipment side of things, do you have suggestions for adequate equipment for potentially larger outdoor applications? So, for instance, in container nurseries.
Yeah, that's the tough thing about larger applications like this. It's just the practical way of doing it. So Yeah, it's honestly it's just it depends on what equipment you have. Um, yeah, I mean there's those big fertigation carts that you can drag around. To be perfectly honest, I don't work with a ton of nursery folks, so I don't know the kind of equipment that they typically have. So I would just have to see what they have and what's just typical in those situations, to be honest.
We got lots of more questions still. Still hope you're doing okay there, Greg.
Not
So,
fun. Yep.
how this person's asking, how long can IJs survive in the media if they cannot find a host? We kind of already answered that,
Yeah.
but I'm not sure if there's something else you want to add.
Yeah. Same answer.
Yeah, stuff. Sorry. Having trouble keeping track. All right, what about these? Would these be compatible in a prop house where URC are being dipped in EpiShield?
Oh, for sure.
Awesome. Um,
Yeah, because
okay.
Greg, I think you mentioned at your CGC talk that you can mix nematodes with pretty much anything without killing them.
A lot of stuff, yeah. Like organophosphates tend to kill them. Some of the really heavier pesticides aren't so good for them. And obviously, like you wouldn't want to use like a nematicide at the same time. But yeah, they're compatible with a lot of different pesticides. Like it's hard to kill a nematode. Like you've got to do it on purpose, pretty much.
Um, this one's a good one. So I understand some species of trichoderma may be antagonistic against root knot nematodes. Have you seen this antagonism occur between any trichoderma and beneficial nematodes?
No, so that's interesting about the root knot nematodes, but I mean root knots are literally making nodules in the roots and like feeding in the roots. So I can see if those roots are being occupied by a fungus living on them, that might impact them. But the entomopathogenic nematodes, they have nothing to do with the roots. Like they don't care about them at all. So it's totally fine to use them at the same time.
So, we have two questions here about the symbiosis with the bacteria. So, essentially, can the bacteria survive without the nematodes in the soil, or do they fully rely on symbiosis? And if so, can the bacteria be found commercially, just be sprayed alone?
Nope. So that bacteria cannot survive in oxygenated environments. So it can only survive within the nematode and then also within the insect cadaver. So just like every other stage of nematode except the infected juveniles, the bacteria cannot be free living.
So, all right, there's a question here about size difference of EPNs. Could that be why there's different activity against Parvis vinus? Sarah, I don't
That I'm not really sure.
know.
Um,
Sure. I don't know. Are there size differences between the nematodes? I mean, obviously the thrips, like parvi larvae are really
Regularly.
tiny. So like compared to Western flower thrips, so it could be something to do with that. But I mean, technically only one infective juvenile needs to fit in there to
Right.
kill them. So
Exactly. So yeah, I've actually never even looked into how big each nematode is compared to other species. So I'm not
yeah.
really sure, to be honest. It's a good
Every time
question.
we find a new pest species, it opens up all of these other biological questions. And
Right.
now it's like another rabbit hole of, yeah, like why doesn't this work? So I
Yeah.
think that'll come later. I think with Parvy, we're in the like, let's just see what works, and then we'll figure out why it doesn't work
Yeah,
later.
exactly. Just get rid of it. We don't care how.
And I think this had something to do with, sorry, about the results that you're mentioning, Sarah. Someone just mentioned, could Alex's results be based on them having higher temps in their trials?
Oh, for sure. Because like they're doing their research in like research greenhouses in Florida. So again, I think hopefully that paper will shed some light on what they were doing. So like I said, I will link that on the blog. And someone else I noticed in the Q&A asked if they could have Greg's slides. So I will probably invite Greg to write a summary blog post of all of this that we can keep on the blog. And if not, the recording will be available on the blog or the On Floriculture YouTube channel.
Awesome. And then there's just some last few comments in here. Oh, there's one more after that. But Julie just mentioned some comments about studies done at Cornell on nematodes controlling fungus gnats. So there's a link that she's put there in the chat. And also said that nematodes can be applied through drip tubes, but some things to be aware of when doing that. I suggest working with someone the first time you apply them to outdoor nursery irrigation systems to ensure everything goes well. So it's great. And then there were just some comments here too about size. Yes, there's differences in nematode size regarding the prior question. Carbocapsy is definitely smaller than Veltier, so maybe it can fit into the parvis better.
I really just
Um,
now want to see a bunch of videos of nematodes trying to fit inside puppy
Yep.
larvae.
Yep.
Or like the pupae, I guess, but they would also be smaller. So yeah, anyway. Yeah,
and
I
then
just
there,
wanted, is there
oh,
any more
sorry, one
questions?
more question that came
Okay.
in, and it's a good one. Why don't nematodes harm beneficial insects like hypoaspis?
Well, so typically mites are too small for nematodes to get inside of. So hypoaspis or stratolaelaps. In fact, it's kind of the opposite with them. With that specific example, stratolaelaps actually like to eat nematodes.
Yeah, I think the only beneficial insect they can really affect is a theta, a Delodia, which
Yep.
I only know because I did my master's on that and did videotape nematodes going inside a theta, but it was really low levels overall, like under 15%. So they just have their preferred hosts that they've evolved with. So
And it just
um,
happens to really work out for us because they like to go after the pests.
yeah, exactly.
On
Well,
that
thank you.
note, do you think they would be able to infect beneficial lacewings?
It's not found
If
in the same
they're
areas. Like,
if they're in the right spot.
yeah.
Yeah.
Well, with that, I just wanted to thank Greg for giving this talk not once, but twice, once at the Canadian Greenhouse Conference and now through a webinar. We should all be thinking about using nematodes more, especially on spring crops and propagation, where we know we get a lot of fungus gnats and shoreflies and other fun things. So with that, everyone have happy holidays if the GrowON team doesn't see you before then. And thank you so much, Greg.
It's fun. Thanks.
Thank you.